My name is Julie, and I am a fifth year, as Dario was describing, like at Caltech, and I'm going to present our work DeepCell Types, a generalized self-phenotyping model for spatial proteomics with language-informed vision models. I know there's a lot of terms to unpack here, but I would just like go through them one by one. So the outline is pretty simple, like the classic outlines you would see with like talks like this. We're gonna start with backgrounds and challenges, and then we'll talk about our approach, and then a little bit results. And so this work is mostly on spatial proteomics, and I think in the recent years, multiplex imaging in this field has been pretty promising. So there are a handful of ways that you can use to capture the protein markers simultaneously. And let me remove this. So the number of protein targets you can capture usually ranges from like 10 to even 100. And this gives you a lot of information about the tissue and elucidates a lot of like mechanisms in terms of like human disease and treatment. However, this technique generates very challenging data. So you usually get a dozens of images as a stack, and each channel represents a protein marker that you're trying to capture. And there are usually like thousands of cells in one single image. And the first task is usually to do segmentation on these images. So you usually want to capture where the nuclei is and where the cell boundary is. And after you crop out this small region corresponding to one single cell, then the next step is to identify the cell types. So you would have like a bunch of crops like this, and you would have their marker names corresponding to each small crop. And then using this information, you will decide, okay, what marker exists and what doesn't exist. And then you infer the cell type from that. So my work mainly focused on the second step, which is self-phenotyping. But in our lab, we also developed a bunch of tools for segmentations as well. So namely, there are MASPR, which is mostly for tissue phenotyping. And recently, we released CellSem, which is a foundational tool for more general purpose cell segmentation. And they're both available like online. You can just drop your image and try to use these tools for cell segmentation. And so going back to my work, phenotyping, there are a bunch of challenges with this task. So the first is what we discussed previously, like there are a bunch of ways to image the cells. Therefore, you have a variability of imaging modalities. And secondly, like there's also the variability of artifacts and noises. Sometimes you get a segmentation that is just not optimal, and sometimes you have noises from your tissue. Sometimes you have like spillovers across cells. Sometimes you just have like cellular projections that exist across cell boundaries, and that's something can make the self-phenotyping task pretty challenging. And I think the biggest challenge is actually the variability of marker panels. So I think each experiment would target a different, like, for example, disease or tissue, and therefore scientists usually design their marker panels according to their needs. So one experiment will have, like, for example, a marker panel that has steady 163, and another might have like a completely different cell panels. And then a lot of markers might not overlap. So to build a model that can generalize across these different marker panels is quite challenging. And I think prior to our work, there was no such model that can actually achieve automated and generalizable solution agnostic to marker panels. So we pondered on this problem for quite some time because it's it's a little bit daunting, like how you can actually find a model invariant to the input set. And when you think about it really closely, you realize that this problem is very similar to a language task. For example, like you'd have two sentences, and a simple task would be to like determine the sentiment. And for those sentences in language, you usually also have pretty variable lengths. And like once you draw this connection, then we decided to just like borrow some insights from language modeling. And I think the recent there has been like a recent surge of large language models, and under these successes, the architecture of transformer was pretty important and helpful. So we wonder if we can just apply the mechanism of transformer to the spatial proteomic task. And so with a sentence, if you were to determine the sentiment, it usually works like this. You have a model that learns the uh, correlation between these tokens. And then you will have like this learnable class token that is like basically a vector, and you classify the token in order to determine the sentiment. And all these like arrows here means like the correlation between these words. So for example, like in this sentence, the word very and boring is super important for the sentiment determination, and all the other terms are less important. And if we draw this correlation to our self-phenotyping task, we're basically asking whether we can learn the correlation of these tokens, which represents the markers, and then learn this class token, which we can further use to classify the cell types. And maybe in this case, Ki67 or CD133 could be super important for this cell phenotyping. This is just like a mock example, but you get the idea. And so taking this idea, we want to build this channel-wise transformer, which is agnostic to marker panels. So if this model is trained on this first row of markers, then it can generalize to a second row of markers. And if you compare this to the more traditional approaches like maps or cell-siders, which are either NLP-based or convolutional neural network-based models, they usually have a fixed number of channels. And if it's trained on the first row, when you apply it to a new dataset, then what you would do is, for example, zero fill the missing channels, and it can never take this new channel into as input. So I think this mechanism enables us to build this more generalizable model that's more agnostic to the marker panels. And going in a little bit detail into this model architecture, we have a channel-wise transformer. We also have this language-informed vision model part. So these are the input to our model. We have, as we saw before, like some crops of cells, and each channel you have the protein marker expression cross like that varies across channels, and then you have their marker names associated with them as well. So like both this image and this text representation of the channels are going into our model. And on top of that, we also have some more spatial features, which includes a mask corresponding to the cell itself, and some neighbor masks basically tells us like what are the cells that's adjacent to the cell of interest. And there's also the distance transform which tells the model like where is the cell boundary and where's the center of the cell. So taking this, we pass like these three separate things into three encoders. The first batch of images goes into this per channel encoder, which is just like a regular vision model. And the texts of those markers goes into this prompt. So it for example, if we have ECAT, then we will have like a paragraph describing what ECAT is and what cell types it typically represents and its functionality and stuff. And then we send it into this marker encoder. And so the third part, like this spatial information, also goes into like a typical vision module. It's like a convolutional neural network stuff of things. And then we combine the per cell embedding and the spatial embedding so that they give us like the aggregated uh, vectors. And so each vector represents to this the spatial information of this channel. For example, the first one is ECAT, the second one is PanCK and stuff. And here we also have the text vectors, and each vector here also corresponds to the textual information corresponding to that channel. And then we would have this channel-wise transformer, which, as we were describing, like it learns the correlation between those channels. And then it also has this class token, which we can use for the phenotyping. And one additional module that we find useful is this mean intensity projector. It works basically like a residual connection. So when we're learning these spatial patterns, like because these encoders are trained like per channel, it doesn't necessarily preserve like the relative intensity very well across channels. So it turns out that if you have like a mean intensity calculator and then you project it and then combine it into the later layer, it works pretty well. So this would give us this learnable class token and these integrated per channel tokens. And for this task, we have like three learnable heads. The first is cell type head, which is like a MLP layer that gives us the cell type classification result. And the second is a marker positivity head. So basically, for each of these channels, it can tell us whether it's positive or negative. And we also have this third hat, which is a modality hat. And so it's actually an adversarial task. So the forward pass, we're trying to have the model predict the modality, but when we're trying to like pass a gradient back to the through the network, we want we reverse the gradient so that it unlearns the modality. So the idea here is that because across different modalities, there's always some variance like that's systematic to those imaging modalities, and we want the model to be more invariant to that. So we want the latent representation of the model to unlearn this modality. And we have some like latent space analysis, which I will show later, that prove this to be useful. And on this slide, I also want to dive a little bit deeper into the two specific components. So the first is this marker encoder. So take each of those protein markers. We first like generate this prompts, and then it goes through this frozen embedding model, which we're using GPT embedder. And then we have this SVD step, which condense the I think it's 4,000 dimension embedding into about like 500 dimensions. And then we have this learnable linear layer that generates this marker embeddings. And the other component I think people ask a lot is like, what is this marker positivity head? So after we pass this embeddings into this marker positivity head, like we actually learn a shift plus scale operation on top of that. So this for basically for each marker, we learn these two layers that determines like how we should scale and shift a marker so that like the threshold for determine whether a marker is positive or negative is modulated by the marker itself. And the output of this layer is a logits that goes from 0 to 1 and tells us whether the marker is positive or not. Okay, I think that's a little bit like too much detail on like the model itself, and let me we can talk about these parts if like at the free discussion session. And so with that model, what we did next is to curate a bunch of data. So we developed this DeepCell type graphic user interface so that like an expert can look at the images and label the cells. And so with that, we curate a very large and diverse data set. We have about 9 million of cells, and which covers like 7 imaging modalities and a bunch of tissue types, and also across cell lineages. And here is like a hierarchical cell type graph we have. And unsurprisingly, like the cell type counts is a little bit correlated with imaging modality. For example, astrocytes only comes from IMC, and lymphatic endothelial cells only comes from codex. And there are some like very unique cell types that are only from IBEX. So that is like part of the reason why we wanted this adversarial module, and also like if we can like our goal is to keep curating an even bigger data set so that like the correlation between the cell types and imaging modality can be alleviated to some degree. And we're in collaboration with like HuBMAP, which is a pretty big consortium here in the US. And we are collaborating with like about like 40 universities to like just label more and more more cells, yes. So we were able to show that our model can have pretty robust performance across tissues and imaging modalities. And here is a quick visualization of like across different tissue imaging modalities and tissues, like the model is able to do decent predictions. And here is the confusion matrix. And if we compare our model against CellCider and MAPS, we see a relatively big boost of performance. And the other interesting fact is that if you pre-train this model and then fine-tune it, it doesn't give much performance boost compared to just like train supervisedly from scratch, which I find a little bit surprising. I think given our time constraints, I'm gonna just quickly go through these slides, and we can discuss more later in the free discussion session. So this is the latent space that I was describing later. Earlier. Like the idea is to mix the imaging modalities in the latent space so that the model is not learning the artifacts that introduced by different modalities. And by visualizing the latent space, we're able to show that like it is more organized by the cell types, and within like the same cell category, like imaging modalities are relatively good, well mixed. And so this plot basically shows that model confidence is well calibrated. So the model has like a predicted probability, and there's also the observed accuracy you can calculate post hoc. And if the model is 100% like perfectly calibrated, then when it says 0.8 confident, it should give about like 0.8 accuracy. And what we observe is this solid line here. So when the model says it's only 0.8 accurate, it's actually about 0.9 accurate. So in that sense, like the model is relatively more conservative, and we can trust its prediction a little bit more. And of course, when the like predict probability is like below, for example, 0.4, you wouldn't want to trust the model's prediction and would just label it as unknown. And the next result I want to talk about is the marker positivity threshold. And as I was showing earlier, like there was the per marker modulation module. And what we see is that like compared to like a fixed threshold, which is like usually 0.5, if you allow each marker to have a different threshold, what you see is actually a distribution like this. So some markers, the majority of markers are around like 0.5-ish, but some need a really high threshold and some needs a really low threshold. So that kind of comes from like the variability of like the marker stance. Like some are just darker and some are just like dimmer, and the model is able to adapt to that. And if we compare our marker positivity prediction against like a solar method, which is Nimbus, what we see is that on the Nimbus dataset, like to give a little bit more context here, Nimbus is also like a pre-trained model, and so is ours. And so if you train so Nimbus is trained on their own dataset, and if you run the marker positivity prediction, they perform pretty well on their own dataset, but they perform much worse on ours. And if we do the reverse experiment where we predict on our dataset, our model performs better, whereas Nimbus performs worse. So I guess the conclusion here is that like there's certain bias that the model itself cannot overcome, and the pre-trained model often performs better on their own datasets. And I think this is the last slide. So here is a quick comparison between the classic pipeline versus our model DeepCell Types. So typically, like given this multiplexed images, after segmentation, you would calculate the mean intensity and do some sort of clustering, and then you need an expert to map the categories to identify the cell types, which is pretty manual and time consuming. And according to our collaborator, this whole process usually takes a couple months. And whereas like if you use DeepCell Types, it is automated and you can get the results within like 10 minutes, to be generous. And so if you apply this to an actual FOV, this is ground truth, and these are the predictions. And I think the dark ones here are the error predictions. And I think on the classic pipelines, a lot of those are just unknowns that the experts are not quite confident of. And with that, I'd like to thank you for your attention and thank all my collaborators and our funding sources for the support of this work.